I'm Pathmanathan from Beacon Biosignals. I'm going to talk about a little bit about what we do once I get these slides to work. There we go. I am an employee of Beacon Biosignals, as you can see, so everything I say here is biased. Don't trust me. This project is funded by a consortium of industry sponsors with whose efforts were immensely valuable, and this couldn't have happened without them. And definitely the efforts of Cure SYNGAP1. I'll say one thing about that, and mainly the patients and families living with SYNGAP1 mutations. We originally had intended to do, I forgot what the exact number, our target number was, something like 20 participants, and we recruited that number in like a month. It was astounding. The organization of Cure SYNGAP1 is just off the charts. And so we said, okay, well, we think we can get more headbands, so let's extend this trial. So it's wrapping up soon. It's still ongoing, so I'm just going to present some preliminary data from this home utility and feasibility trial. So really what I'm going to be talking about is the waveband EEG, which I'm holding in my hand. It's got I'll talk about in detail in a little bit. It's got 4 EEG electrodes, and the beauty of it is that's it. I'm recording EEG, right? And it streams data to the cloud, and then that data is sleep staged, and it's FDA cleared for adults, not FDA cleared for children yet, but we're working on that. And the data quality is very good, but it is very sparse electrode coverage. That's the trade-off, of course. There's no tech involved. There's no skin abrasion. There's no prep. They're dry electrodes. And our question was, is this data useful or valuable in SYNGAP1? And I'm going to talk about that in the context of what we know about EEG biomarkers, some recent, in my opinion, amazing advances by the CHOP team, and then some future directions that I think hopefully this will add to. So if we look at the patient journey, this is a figure from NORD. It's a mess. Like, this is just chaos here, right? And the families in the room will understand this better than anyone. It's a slog to get to, and here you're only getting to a diagnosis at the end of that slog, right? It's a complex journey. It's prolonged. It really is an odyssey, right? And in just one little figure here, there's like specialists and a test, right? Like this little stethoscope down here. Sorry. You can see the stethoscope there, right? That's supposed to represent all the tests, and those tests include some, I mean, they're not invasive necessarily, but annoying, hassle, difficult tests to obtain, including EEG. And in the odyssey, Poseidon is really the one who's the antagonist, really holding everyone back. And you can imagine that Poseidon is the EEG, right? Like, it's just EEG is a difficult thing. It seems so easy to me. Oh, I got a screen full of data. This is great. But for families, this is a difficult thing. So when we ask for EEG data, that's a slog. And that's too bad, because there is information in the EEG. We know that many patients with SYNGAP will have a variety of EEG abnormalities. So here, over here, I'm showing you slowing. It's actually slowing. This is the left hemisphere. This is the right hemisphere. Even if you don't know how to read an EEG, squint, and you can see that there's a difference here between here. There's these slow oscillations. That's slowing, right? And if you look at the back portions of so here's the posterior segment, these frequencies aren't what I would expect. So I can just eyeball that and say that this is abnormal. And those are abnormalities you will see in SYNGAP. You will also see interictal epileptiform discharges. This is the hallmark of epilepsy. And if I saw that, if an EEG, by the way, that data at the bottom there is from our headband, from one of our patients in this trial. If I saw that, I would immediately say, yes, you have epilepsy. I mean, that should not be on anyone's EEG. If you have that, either you have epilepsy or maybe a very close relative has epilepsy. Now, that you do see it in a very rare subset of people who will never have a seizure, but that's very, very uncommon. These features, as you've heard throughout the day today, they're not static. So if you have them at one point in time, you don't necessarily have them forever. They do change with age, and the distribution changes with age, which is really important for us, because remember, we don't have a lot of electrodes if we're going to use this headband. We only have frontotemporal and occipital, which in SYNGAP might be okay, because that is a common area for discharges to be present, but we just need to prove that we can identify things. This is a paper from Dr. Sheffer's group. eyelid myoclonia, you've heard a lot about that today. Absence with eyelid myoclonia. That is a tough thing to see on regular EEG. And one of our questions was, can we pick that up on the waveband? I'm actually going to, spoiler alert, I'm not going to have the answer today because it's a lot of EEG data. We're going through it. I'm not, but team is going through it manually. And remember, each patient has many nights of data. So it's being reviewed. I have looked at quite a bit of the data. I don't know that I can see eyelid myoclonia on our electrodes, but I'm happy to discuss that later. So there's a lot of EEG features, and we've been talking about EEG biomarkers for a long time. What are we going to use them for? What could they be used for? And the answer is, well, we can definitely use them to help in diagnosis, but we're still working on, can we use them to track disease progress or treatment effects? And I don't know that we're quite there yet, but could we be? And I think the answer is yes. And I'm going to go back to Dr. Scheffer's paper, and actually she's already talked about this quite a bit. Dr. Levine talked about this. I'm going to point out some of the comorbidities in SYNGAP1, and I'm going to focus on one which I think you probably already know what I'm going to focus on because Dr. Levine already pointed out, sleeping problems, right? And I don't think I need to dive into that much more because Dr. Levine already covered this in excellent detail. But I will say that sleeping disturbances are very common, and that's critically important to understanding epilepsy as well as many other neurodevelopmental abnormalities. In epilepsy, sleep can activate IEDs, activate seizures. Transitions between sleep states are very informative about deep neural circuitry, hypothalamus, brainstem. So sleep provides an amazing window into cerebral function that you don't otherwise get in any other way. And EEG is a wonderful window into sleep. So as has been pointed out, this is a new area of interest. There's not a tremendous amount of data, but here's a nice view in rat EEG, which I don't know how to translate into human EEG, but the thing I take away from this is there are absolutely spectral differences in the EEG. So the frequency content of EEG is quite different in rats with heterozygous for a SYNGAP mutation versus wild-type mice. So in this figure, the blue line here is a SYNGAP mutant mouse, and then the black line is wild-type. And what you can see is it's actually stage dependent. So there's not a huge difference in REM, at least in the rat. That may or may not be true in humans, but in the rat, REM sleep looks very similar, but in non-REM, which is actually they're consolidating a number of stages together, sort of simplifying it. I don't know if rats do have all of the stages we have, but I don't know how easy it is to identify them. But when you do consolidate those stages together, you'll notice a dramatic difference to the point that I wonder if you could actually use that difference to classify patients. Is this someone with SYNGAP versus not? And that's one of the questions we're gonna be asking. So we have done this kind of thing in other disorders, not SYNGAP1. Notice the title of my slide. This is not SYNGAP1. This is a sleep disorder. And if you look at EEG features in sleep, there's a countless number of EEG features. If you just look at spectral features by stage, we pulled out 720 prominent spectral features. That includes things like absolute frontal alpha power, relative frontal alpha power, alpha theta ratios, all these things that if you follow EEG literature, you've heard these terms before. There are a vast number of features because you have features for every channel, and then you have features for every spectral frequency, and then you have to multiply that by the 5 sleep states, wake, all the NREM states, and then REM, right? So you wind up with this massive array of this massive feature space. And that's just the spectral features. You can also talk about micro sleep features like spindles. And in our paper, we did look at spindle features. You can look at macro quantities like total sleep time or the amount of time spent in REM. And you wind up with an immense amount of data, and you can feed that into AI models and build classifier models. And in a sleep condition, actually a sleep condition in which EEG was not thought to be helpful. This was actually insomnia, in which EEG is actually not used for diagnosis currently and is not thought to be helpful. We were able to build a classifier that had incredible accuracy in determining not just whether you had the condition, but which type of insomnia you had. So it's definitely useful. Now, okay, that's a sleep condition. You may not believe that insomnia can't be classified by EEG, which it is not a well understood thing until we wrote this paper up. But guess what? People are doing this same concept in Syngap, in the DEE space. So our colleagues from CHOP who are in this room, I think they're gonna be talking about some of this data in a moment, so I'm not gonna steal their thunder, but this is amazing work. They took spectral data, so here you can see Syngap is the blue bar. You can see that there's a difference at certain age brackets in the alpha theta ratio, so alpha delta ratio. So one spectral feature does distinguish at different ages between a variety of DEs and neurotypical controls. When you take a whole bunch of features, not just the alpha delta ratio, and I think that they used something on the order of 50 features, but I could be wrong, and I apologize if that's not correct. But it was a large number of features. They were able to, number one, distinguish between neurotypical and Syngap, or neurotypical and STXBP1, with very good accuracy. And here are the top, I think, 7 features that that went into their model. But the other thing that they found, which is really amazing to me, is they were able to characterize, or they found an association. I shouldn't say, they weren't able to, you can't use the word predict here, but they found an association between spectral data in the EEG and motor outcomes. And it was stronger than age. And age is an incredibly strong predictor of motor outcome. Oh, you have a 2-year-old, should be walking, should be running. EEG was actually better than that. That's just a tremendous finding to me. This shows you what could be done. And they have an amazing data set, and it takes a lot of data to do this. And so how do we increase that data set to build these models? So what I would say about the work that CHOP has done is this quote. This was actually a quote by the reviewer to our paper in insomnia. And it was just so eloquent. And I've never had a positive review like this. This was just mind-blowing when we got this statement back. But what our reviewer said was, these findings move the field beyond the binary question of whether, and I'm going to insert SYNGAP1 here for the CHOP team, has an EEG signature. It does, but that signature is distributed, dimensional, and heterogeneous. The value of this EEG-derived score or model lies not in diagnostic substitution, but in clinical augmentation, stratifying patients, sharpening trial design, and steering treatment choices. And that is absolutely the goal. That is, I think, what we'll be doing with I won't say EEG biomarkers, I will say EEG-based models, because it's not going to be one feature. We're not going to be looking at just alpha theta or just spikes. All right, so our perspective is EEG data will provide incredible diagnostic information. It's not just going to be specific microfeatures like discharge. It's going to be spectral data and it's going to be multidimensional aspects of the EEG. And we will build models that are very informative, but Poseidon with his EEG, it's difficult to get that data. The data that CHOP used was 1020 EEG. That's a hard thing to get. Could we use something like this headband? And here's Penelope, our heroine, with what ChatGPT thought was waveband. We got to work on that. Could we, therefore, use a device like this to collect data longitudinally, and not just when a child comes to the hospital, not just when someone might be sick and you're otherwise ordering an EEG? Could we use this to gather data weekly or at any point? When a child is doing really well, could we gather data? So really quickly, I suspect I'm way over here. So this has, as I mentioned, 4 electrodes, as shown here, F7, roughly F7, F8, O1, O2. It currently, in the next iteration, we will have pediatric sizing. Currently, it is one size fits all. It is adjustable. It's got a stretchy band here. At the smallest size, it's about 50 centimeters, which is roughly 50th percentile for a neurotypical 6 to 7-year-old male. So it's not going to work for every child, unfortunately, and that was a limitation. So what we wanted to do is we wanted to have, instead of the Poseidon version of EEG, we wanted to gather data at home. This is also ChatGPT figures for me, where ChatGPT actually did get a nice waveband this time. Wanted to have parents gather data, or caregivers gather data from their family members living with Syngap, and then see what see whether they could actually operate the device, whether their children and adults, we included adults in this, could tolerate the device, and whether we could do this over multiple nights. And we actually had done this last year in LGS, and we got 10 out of 12 participants with LGS to complete the study, and all 10 neurotypical siblings. And we were able to capture seizures, IEDs, GPFA in participants with LGS. So the study structure is fairly simple. It's a total of 10 nights over 2 weeks, number of questionnaires, and fully operated by parents without any intervention by a technologist or us. And many of the families in this room actually participated in the study. Thank you again. And then it is mainly a usability feasibility study, so our main objectives were just to see if we could gather data. But then we do know that data is going to be useful, so I'll present a little bit about what we found. In summary, the study is still ongoing, so this is very preliminary, and I lost a lot of friends in my engineering and data science team by beating them over the head over Thanksgiving to get me some of this data. So this is truly hot off the presses. So all the subjects recorded some amount of EEG, most of them at least a routine clinical EEG duration, most of them hours long, and then a majority multiple nights. I think the average was 5 full nights of recording. So definitely usable by parents, and not every child, but the majority of children tolerate it. Interictal discharges were captured. GPFA was captured. I alluded to this earlier. I am not the one reviewing the raw EEG, but I have looked at a lot of it. I don't know that I've been able to identify eyelid myoclonia on this data yet. But again, to be determined. It does automated sleep staging, and there are, we're going to need to dive into this, but there are definitely patterns that distinguish children with SYNGAP1 from neurotypical controls. And then the spectral data. Really briefly, just a couple of interesting things that pop out. So as has been reported by others, indeed there is increased delta in wake and decreased alpha. So you lose some of the faster frequencies, you gain some of the slower frequencies, but it's state specific. It is absolutely state specific, and most people when they're looking at EEG, they're only talking about wake. But if you look at other sleep stages, the pattern is very different. And I wonder, I don't actually know the answer to this, but I wonder if, so it may be that many conditions have increased delta and decreased alpha, but does the pattern hold in their sleep states the way it does here? Could I use this figure to distinguish between SYNGAP1 and STXBP1, for example? I actually do know that STXBP1 does have a slightly different pattern. The slowing is persistent through many other sleep states in STXBP1. If you look at across ages, these spectral differences aren't stable, so they change over time, and some of them are actually very dramatic. This is a very strong finding. I mean, again, preliminary data. We haven't included all of our participants, but frontal beta it separates as with age. So it's actually children with SYNGAP have elevated frontal beta relative to neurotypical neurotypical children. And that difference gets stronger as the kids get older. So I think that the band is usable. Certainly it's usable by caregivers. I think many children are able to tolerate it, not all, but many. I will note that even though we're not done, I think that the rate of usability is going to be slightly lower than what we found in LGS last year. But still, a majority of our participants were able to record multi-night data. We are able to identify epileptiform features, but one caveat, this is not a head-to-head against 1020, so we don't know what we're missing. And clearly, we must be missing things, right? We only have 4 electrodes here. And the spectral abnormalities that have been described previously are detectable and measurable based on the data from the band, and they do differentiate from neurotypical participants, and there is an interesting state specificity. And so what I would envision is in a near future world, we use all of this data, and not just wake data, to refine the models that have already been created that I think will provide great insight. And my hope is that could give you a a quantitative assessment of where someone is with their disease process that could actually be used to monitor treatment effects. That's actually, I think, where we could get to, although that has to be proven. All right, with that, I will stop there. Again, I would like to thank the consortium, but especially Cure SYNGAP1 and all of the families that participated.
Thank you.